Hello everyone. So today we are going to see what is the basic concept of neurotransmission. So before going on to the actual process, let us see that how the action potential travels. So this is the you have the axon end. So this is the presynaptic terminal. So this is the synaptic knob. Okay, so the synaptic knob will contain many synaptic vesicles. These synaptic vesicles are filled up with the neurotransmitters. So here we can see that the neurotransmitters are actually traveling from the, so this is your cell body. So these neurotransmitters are synthesized here and then they are packed into the synaptic vesicles. These synaptic vesicles, they travel down to these axon. Okay, so this will travel to the down to this axon. So once it has reached the synaptic knob, now whenever the stimulus is reached and this axon undergoes depolarization, so we will be seeing the concept of depolarization on these things. So whenever the depolarization happens, then these synaptic vesicles, they will fuse to the membrane with the membrane of this axon, and the neurotransmitters would be released in the synaptic cleft. So what is synaptic cleft? It is the region between this presynaptic terminal, okay, or your presynaptic membrane of the axon, which is carrying your signals, which is carrying your neurotransmitters, and this is your postsynaptic membrane, which is a part of the dendrite of the next neuron, where the relay of action or the relay of the neurotransmission is going to take. So this postsynaptic membrane contains many different your receptors. So these receptors are specific for the neurotransmitters that are being released by this synaptic vesicles into the synaptic cleft. So these neurotransmitters go and bind to this receptors at the postsynaptic neuron, and then whatever the action that is going to be there, that is carried out. So most of these receptors, they are bound to channels. Okay, so they are your ligand gated channels, so where your neurotransmitter is acting as your ligand. So the transmission of the nerve impulse along your nerve fiber can be summarized in 3 steps. So one is polarization, which is also called as the resting potential. Next is the depolarization, which is the action potential. And next one is your repolarization. So we will be seeing all these 3 concepts. So the resting potential of any neuron. So resting potential, that means it is not doing anything. So that is minus 70. So why it is minus 70? So if you see the exoplasm, so this is the exoplasm. So exoplasm, it contains more of your potassium ions. So the potassium ions are around 25% more as compared to outside of your membrane. So if you see this particular picture, so in this we can see that these brown ones, they are your potassium ions. So this is the inside of your axon. This is the blue part is representing the outside of the axon. So here you can clearly see that the concentration of the sodium ions are more inside the axon and it is less outside the axon. And your sodium ions, on the other hand, it is opposite. It is well less and in the inside of the axon, so that is in the exoplasm, and it is in higher concentration outside the exoplasm or outside the membrane of the axon, that is your exomembrane. So whenever your neurotransmitters go and bind to these receptors, most of the receptors are your sodium ion channels. So sodium ion channels open up and the sodium ions will start influx. It will come inside the cytoplasm or your exoplasm. And that's how, now what happens is, because more sodium ions, so around 10 times higher of sodium ions, now influx inside the from minus 70, the potential now moves to plus 30. So because of positive ions coming inside. So we can see over here. So this process is called as your depolarization. We can see because of the opening up of your sodium channels, the sodium ions are moving inside. More and more of your sodium ions are moving inside. Now, as we see in this particular picture, because of this depolarization in one area, now there are various different voltage gated channels which are present. So these voltage gated channels, they sense the change in the voltage that is being caused because of the incoming of or influx of your sodium ions. So because of that, these channels also get activated and more sodium ions are going to now come inside. Okay, so it is a kind of your wave that is going to pass from that dendrite, so which is your postsynaptic zone, till the end of the axon. So along the length of the entire nerve fiber, this wave of depolarization is going to be carried out. Okay, so that is the depolarization part. Now next is your repolarization part. So once it has, most of your part has become depolarized, or the potential has now changed from -70 to positive end. So what happens is, now slowly these sodium ions channels will close. Okay, because they are voltage gated channels, right? When voltage changes, again it will sense that and it will close up. So when that closes, now what opens up is your sodium potassium pump. So sodium potassium pump is going to take up sodium ions and put it outside, and in exchange it would be taking up potassium ions and put it inside, and that too 3 sodium ions are put outside and in exchange 2 potassium ions are put inside. So this will again bring back the whatever the potential of the resting neurons are there. So that was -70. Again that -70 potential is going to be put back. So this entire action potential, that means passing of your depolarization from the cell body, from the top of the dendrite till the entire length of the neuron, this entire part is going to be possible only when the stimulus is very strong. So that threshold of the stimulus has to reach, then only this entire process of action potential will start. Thank you.